Hey there! Welcome to today's lesson on genetic engineering. This is one of the coolest fields out there, allowing scientists to tweak DNA for all kinds of benefits: medical, agricultural, even industrial. Let's dive into the tools and techniques that make it all possible. First up, the basics. Genetic engineering is all about changing an organism's DNA to introduce new traits or figure out what genes do. It's like biological tinkering. The main steps: cutting the DNA, inserting new genes, and then transferring that modified DNA into a host organism. Simple, right? Now let's talk about restriction enzymes. Think of them as molecular scissors. They cut DNA at specific sequences. For example, E. coli snips at GAT-C. This action helps isolate the gene you want to work with. Next are linkers and adapters. These are small DNA pieces that make sure your new DNA attaches correctly to vectors. Linkers help attach foreign DNA, and adapters have sticky ends to join pieces cut by different enzymes. It's like putting together a DNA puzzle. Then we have cloning vectors. These are like delivery trucks that carry DNA into host cells. They need to replicate inside the host and carry the desired DNA. The main types include plasmids, small circular DNA in bacteria; phages, viruses like bacteriophage lambda that infect bacteria; cosmids, a hybrid of plasmids and phages for cloning large DNA pieces; transposons, jumping genes that move around chromosomes; shuttle vectors, can replicate in both bacterial and eukaryotic cells; expression vectors, help produce proteins from foreign genes. Let's move on to rDNA technology. This involves key steps: gene library, a DNA collection representing the whole genome; insertion of DNA into a vector; restriction enzymes cut the desired gene and DNA ligase glues it into the vector; transfer into bacterial cells. Techniques like transformation, electrophoresis, and heat shock help transfer DNA into cells. Selection and screening. Select cells using antibiotic resistance and screen using methods like blue-white screening and PCR to confirm the desired gene. Finally, blotting techniques are crucial for detecting specific DNA, RNA, or proteins. Southern blotting identifies DNA with radioactive probes. Northern blotting detects RNA. Western blotting identifies proteins using antibodies. And there you have it. Now you know the basic tools and techniques used in genetic engineering. Keep exploring this exciting field. It's a key player in modern science and medicine. Don't forget to like, subscribe, and stay tuned for more educational videos.